Good evening, everyone. So, this week we will discuss a topic which has been recommended or actually asked for quite a few times in the past many weeks. We will discuss hair cycle this week. Now, why did I decided to go for hair cycle before discussing hair anatomy? The reason for that is that we have easily gone through melanogenesis, we have gone through keratinization, and last week we did the embryology of skin. In the embryology of skin, we discuss the embryogenesis of hair cycle. Oh, sorry, the hair follicle, and how does a hair follicle develop from a sensitized part of epidermis? Okay, so if we have that kind of development of the hair follicle back in our minds, and we know the process of melanization, and we know the process of keratinization, understanding what all changes happen as hair goes from anagen to catagen to telogen, it will be easier to grasp the various changes that occur. Okay, we will discuss hair anatomy perhaps next week, but we'll discuss the hair cycle this week now so that we can understand and easily comprehend what is happening to the hair follicle. Most of the time, the difficulty lies in remembering the order of what all changes do we expect in a certain phase of hair cycle. For example, what all changes do we see in anagen phase, or what all changes do we see in catagen phase, or something like these. Okay, so we'll go through each and every phase of the cycle, and then we'll discuss a bit about how does the pigmentation happens in a growing hair follicle. Okay, so without much further ado, let's start a discussion this week on hair cycle and basic biology. Let's move forward. Now, we know that hair follicle undergoes hair cycle continuously during lifetime. So we know that the hair follicles develop before birth. It is in the form of lanugo hair intrauterine, and then that's gradually shed off and we start developing vellus or terminal hair. At the time of birth, the terminal hair are on the scalp, eyelashes, and eyebrows. The rest of the body is covered with vellus hair. Okay. Gradually, during the growth and development of the child, some vellus hair will get transformed into terminal hair, namely the area over the pubic region and the beard area in males. So these area will gradually turn into a terminal hair from vellus hair. Now, understand that during the hair cycle, all the changes occur below the bulge region. Okay. Now, what is the bulge region? You have a hair follicle, you have the attachment of erector pili, and you have the origin, or you could say an attachment of the sebaceous gland. Okay. Opposite to the attachment of erector pili, you will have a light bulge here in the hair follicle. And when I'm talking about hair follicle, this bulge is not in the hair shaft. It's in the layer surrounding the hair shaft, namely the inner and the outer root sheaths. Now, this bulge is the area where you have a lot of stem cells. So the area below the bulge is what undergoes changes during hair cycle. The part of the shaft above the bulge is the shaft that is shed out in telogen phase. Okay, and the part of the epidermis above the bulge will remain as it is. All the changes will happen below the bulge. Okay, the upper follicular region, namely the hair, sebaceous gland, isthmus, infundibulum, hair canal, they remain permanent. That means they do not change over the course of hair cycle. The hair length and thickness depends on what is the body side. For example, the scalp hair grows continuously, except you're suffering from AGA like me. But other people who don't have any issues, the scalp hair will keep on growing um, throughout lifetime. While the hair on the body part, they are not that, they don't grow in length. They have a certain length, and after that, they will be shed off. Okay. And they also mimic the general health of the body. We know that disorders like effluviums happen when the body has undergone a major period of stress. So depending on that, what is the general health of the body will have an impact on hair length and thickness. Let's move forward. Now, this is the pilosebaceous unit. If we need to understand what all changes happen in the hair cycle, we must understand, or in fact have a background of pilosebaceous unit. We know that the follicles have nearly developed in 10 to 14 weeks embryonic gestational age. So we have the hair, then the part which goes inside the hair, or the skin, the part that is invaginated below is known as infundibulum. That means a funnel. The isthmus is the neck portion. Okay. from here till the attachment of the erectophilar muscle from the outside. Okay. Now, after that, you have the bulge region somewhere here, and below that you have the body of the hair and the bulb of the hair. And this bulb is the major site where things are going to happen in hair cycle. And this body part of the hair follicle will be the one undergoing maximum changes along with the bulb. Okay. Now, hair have nearly four classes. Terminal is the hair that you usually see on scalp and on the beard region of males. Pilosebaceous unit is seen in axilla and groin, which is mainly the pubic region, and they develop more during the puberty. Vellus hair is on most of the skin, the body part, and sebaceous pilosebaceous unit is on chest, back, face, all the seborrheic areas. Okay, so let's move forward with that background. So let's start our journey of hair cycle. We'll discuss the changes from one phase to another, and let's start first with the phase of the complete cycle. So this diagram I've taken from Bolognia, and it's a good diagram to understand that what actually happens to the follicle at the time of embryogenesis. So we have the dermal placard stage. After placard, we have the hair germ, hair peg, bulbous hair peg. So this is bulbous hair peg, hair germ, and before that there was a placard. Okay. After that, we have the development of the mature follicle. Sometimes the follicle will undergo one cycle of hair cycle, second cycle of hair cycle, and sometimes another third cycle, the birth happens. And here is the first initiation of hair follicle cycling, and the follicle then will go to first catagen. After the two cycles are gone, roughly around the third cycle, the baby will be born, and the hair will enter a circular arrangement of phases, which we know as hair cycle. And a terminal hair will keep on undergoing hair cycle throughout their lifetime. Okay, so it will go first into the anagen phase, which is the growing phase, then the catagen phase or the involutionary phase, and then the telogen phase, in which the hair is eventually shed off and prepares for the next anagen phase. Okay, so one thing to remember is that it's a cyclical procedure, and in a cyclical procedure, if you're in one part of the cycle, you are preparing for the next part. So telogen phase does not mean that the hair will just fall off and do nothing else. During that time, the dermal papilla and the lower portion of hair follicle is preparing itself to enter again in the anagen phase. Okay, in the anagen phase, the hair is growing, but at the end of anagen phase, it is preparing to enter the catagen phase. In the catagen phase, there is programmed organ deletion. Okay, the cells will undergo depletion gradually so that the hair can then enter telogen phase. Okay, so remember, a cycle is a continuous process. We have divided it into phases for better understanding, but remember, it's a continuous process. There is no clear-cut the clear-cut marker where we can say that yes, the anagen phase has stopped and the telogen phase or the catagen phase has started. And when we're talking about hair cycle, we're talking about one follicle. Okay, so every follicle has their own cycle, own speed of cycle, own duration of cycle, and every cycle of the follicle, sorry, every follicle is out of phase with the other follicles. Okay, now what do we mean by that? So for example, if this is the, let's say, the scalp, and you have follicles like this, follicle 1 would be a bit, the cycle would be a bit different from the follicle, let's say follicle 4. Okay. However, 1 and follicle 2 can have similar cycles, in which we say that their cycles have synced. Okay. Now this syncing of cycle is known as quorum effect. That means a follicle can induce similarity of hair cycle of the surrounding follicles. Clear? Now we know that follicle 1 and follicle 4 are different in cycles, and that is known as asynchronicity of cycle. That means the cycles are not going to go together. That is why in humans, you don't go bald for 3 months, then you start growing hair, then again bald for 3 months. That is not we see. We see gradual shedding of about 50 to 100 hair strands every day. And some strands will fall, some strands will grow, some are in anagen, some are in catagen, some are telogen. They are all out of order. That is known as asynchronization. and that is seen in humans. For example, in animals like mice, you have a synchronous cycle of shedding of fur. Okay, so with that in mind, let's move forward. So let's start our discussion with the first phase of the cycle, the anagen phase. The anagen phase is known as the growth phase. Okay, in this phase, the growth of the follicle happens, maximum growth in terms of length, in terms of girth. Okay, the length of the cycle is directly proportional to the length of the hair. Longer the anagen phase, longer the length of the hair. Okay, that is why this, drugs like minoxidil, who increase the duration of anagen phase, promote hair growth. Okay, maximum growth of hair will be there in anagen phase. So growth rates, for example, for scalp hair is about a centimeter a month or 0.37 to 0.44 millimeter a day. Clear? Now in scalp, the anagen phase lasts for about 2 to more than 8 years. This site has the longest anagen phase of all body hairs. At any time, about 90 to 93% of hair are in anagen phase. Clear? The anagen phase for other sites, for example, at legs, it is 5 to 7 months. In arms, it is 1.5 to 3 months. In eyelashes, 1 to 6 months. And fingers, about 101 to 3 months. Okay. So whenever you see an eyelash fall, don't pick it up and wish for something. That's superstition. Just remember that that shaft has just underwent exogen phase. Be thankful for your eyelashes and just blow it away. Let's move forward. Anagen phase has been further characterized into 7 stages. In the first stage, the growth of dermal papilla happens. Okay. Now remember what exactly is dermal papilla. Dermal papilla is nothing but a collection of dermal mesenchymal cells. Now these mesenchymal cells will come together, they will congregate, they will form a collection, and this collection will stimulate the overlying epidermis to form newer hair. Okay. So in the first stage, dermal papilla grows. Mitotic activity starts in the overlying epithelium. So if this is the epithelium and this is the dermal papilla, it will send signals to the epidermis, and these signals will allow the epidermis to undergo changes for the development of new hair. Okay, so new hair will start to develop. Doesn't it seem like the same way in embryogenesis the hair follicle is forming? Because there also you have the mesenchymal collections forcing the overlying epithelium to form a hair placard. And after hair placard, the hair follicle is formed. So it is mimicking its embryogenesis. The hair cycle is mimicking the embryogenesis, and that is why I decided to do hair cycle first, then hair anatomy. Okay. Now, in the second phase of the cycle, the bulb matrix cells envelop dermal papilla. Now, what is this? Now, remember, when the hair has shed off, only the part of bulb remains, not the entire bulb. Only a little part of bulb remains, and this bulb will come down here. It will come down and engulf the dermal papilla. Okay. After this engulf, the differentiation starts. For example, the dermal papilla will start to form newer cells from all the stem cells it has. Sorry, the bulb will start to form newer cells from the stem cells it has while it is receiving constantly signals from dermal papilla. The reverse is also true. The growing hair bulb will induce signals in the dermal papilla to form newer blood vessels to ensure nutrition to the growing hair. Okay, so new blood vessels, new nerves will be developed in dermal papilla, more connective tissue will be deposited in the dermal papilla. So dermal papilla and the hair bulb is talking. Okay, they're talking among themselves. Dermal papilla is stimulating the hair bulb to grow and form new cells, which will eventually form the hair shaft. And the hair bulb is talking to the dermal papilla to form new vessels, the new neurovascular bundle. Okay. In the third stage, the bulb matrix cells differentiate into individual follicular component. Tyrosinase activity starts. Now, tyrosinase activity starts means that the cells will now start forming color. They will start forming melanin. And from henceforward, melanin will keep on getting deposited in the cortical keratinocytes. That means the keratinocytes which are there in the cortex of the cells, the melanin will keep on getting transferred from melanocytes to growing hair. And hair will start to get color. A growing hair shaft is colorless. Gradually, after stage 3 of anagen phase, it will start to develop color. Now, matrix melanocytes reactivate. Now they are starting to receive color and hair coloration starts. So till stage 3 of anagen phase, the hair doesn't have color. In stage 3, the melanocytes start to develop the machinery to form color, the enzymes start to develop, and the color formation starts. From stage 4, the color, the melanin is being transferred to growing hair, and you will have hair color after stage 3 of the anagen phase. Okay. In stage 5, the hair shaft emerges and dislodges the telogen club hair. Remember, telogen starts with the club hair, and if it is released, it is exogen, but it may not be released and it will stay there. A new hair shaft will emerge in the stage 5 and it will dislodge. It will push out the telogen club hair. In the sixth stage, a new hair shaft will emerge from the skin surface. So the new growing hair shaft will emerge from the skin surface, and in the stage 7, a stable growth is reached and the hair will keep on growing. Clear? Now, Bolognia mentions around six stages, but in literature, I've even come across 8 stages, but the 7 are agreed upon. So we'll discuss only these 7 stages. Now, this is a biopsy or the histopathology of Anagen hair. We will not go much in detail. It is nothing difficult to understand per se. You have the hair sheath. I'm not going into outer or inner. We'll discuss them in the anatomy of hair follicle. Okay. We have the hair bulb. This is the bulb hair. And these are the all the collection of stem cells which is dividing to form the hair shaft. So HS is hair shaft, and the hair shaft grows in distal direction outside the hair canal. Okay, we have the iris and the outer and the inner root sheath. So this is the outer, while this thin one is the inner root sheath. Clear? Let's move forward. No. We know that the bulb grows downwards, and in the downward growth, collagenesis and proteases help. So in the growing bulb, a growing bulb will need to make way while going inside the dermis, and to remove the cells in its path, it utilizes collagenesis and proteases to break the dermis and grow downwards. Growth factors are increased like in wounding. So whenever you have a wound in your skin, the growth factors will be locally increased to repair the tissue. The same thing is happening in anagen phase. The neurocutaneous and vascular network in dermal papilla is developed. When that papillary neurocutaneous network is developed, you have the restored follicle or a growing follicle, and this symbolizes the end of anagen phase. So at the end of anagen phase, the hair follicle is developed, the dermal papilla is developed, the neurovascular bundle is developed in dermal papilla, and the hair has come out of the skin. Clear? Let's move forward. After anagen phase, we'll enter catagen phase. So in Catagen phase, everything that has happened in Anagen phase will happen, but in reverse order. Okay. So melanin production will stop first, and you will have a proximal white part of telogen hair. So in telogen hair, you can see that this is hair and this part is colored while this part is white. Why? Because melanin color formation has stopped when the hair has reached there. So when the color production has been stopped in catagen phase, because in catagen phase, all the processes that have been started in a growing hair will start to shut down. Okay. Matrix cell mitosis ceases. So matrix cell mitosis means division in the matricial cells will stop. There's a well-coordinated apoptosis of dividing cells. So all the division which was happening and newer cells were being formed, all the cells will start to undergo depletion. The lower follicle will retract and pull the dermal papilla upwards. So we know that at the end of anagen phase, dermal papilla is in close contact with the bulb. Clear? Like this. It has grasped it. After that, in the catagen phase, the hair shaft will be shorter. It will decrease its length. And as it is decreasing its length, it will pull the dermal papilla upwards. And when it has reached the level of bulge, it is known as club hair, like a club. Like you have those old Neanderthals pictures, na? Like a wooden club, a club hair. The perifollicular sheath, that means the connective tissue sheath surrounding the growing hair, it collapses, and then it is known as a fibrous streamer, which is rich in fibroblasts, blood vessels, and collagen. The dermal papilla reaches the level of bulge at the lower isthmus, and when dermal papilla has reached the level of bulge, the catagen phase ends. Clear? Let's move forward. Catagen phase ends with club hair. The hair length shortens to about 10%. Okay, so 90% reduction in hair length can occur. Movement of dermal papilla from subcutis. We know at the end of anagen phase, the dermal papilla is situated very deep near the level or in fact inside the subcutis also. That dermal papilla is pulled by the retracting hair upwards, and this movement is necessary for the follicle cycle. Why? Because this dermal papilla, which has now moved upwards, will stimulate the hair follicle to go back again anagen phase inside in the anagen phase after the endotelogen phase. Clear? So this movement is essential for the follicle cycle. Anagen phase will begin again via interaction with the dermal papilla lying in the dermis. So the same thing that I said, that the dermal papilla is pulled upwards by retracting hair, and this pulling upward is necessary because when the hair will enter again in anagen phase, it requires its interaction with dermal papilla. If you remember the stages when we were discussing of anagen phase, the cycle starts with the dermal papilla stimulating the hair bulb above, and it can only stimulate the hair bulb above when it is near to the hair bulb. If it is far off, it will not be able to stimulate and the hair will be lost forever. Let's move forward. Now, fibroblast growth factor 5 gene may trigger Catagen state, but is not the only gene required. So it was found that if you mutate FGF5, the Catagen phase never starts and the hair will keep on growing. But there have been instances when you have mutated FGF5, still the hair will enter a Catagen phase. That means it is not the only gene required. Multiple genes are required for shift from anagen to catagen phase. TNF alpha and epidermal growth factor receptor interaction are needed for anagen phase continuation. That means for a hair to remain in anagen phase, you require a proper interaction between TNF alpha and EGFR. When this interaction is decreased, the hair will enter into a catagen phase. Okay, so TNF alpha and EGFR are stimulating the hair to remain in anagen phase. When this interaction stops, the hair will go into catagen. Neurotrophin and TGF beta 1 are molecules which can trigger a premature catagen. That means they will start the catagen phase before it is supposed to start. It can trigger, it can initiate catagen phases. Okay, let's move forward. Now, catagen phase has 8 substages. I tried to find out what are the individual stages. I could not, so I decided to explain this easily in the diagram. Last about 2 weeks, regardless of site or hair type. So all over the body, it is around 2 weeks. There is 50% reduction in dermal papilla volume. So the dermal papilla volume will be reduced by nearly half. There will be loss of the connective tissue and the migration of cells, precursor cells. What exactly do we mean by that? That the connective tissue will be reduced in volume and the cells will undergo apoptosis. Melanogenesis is downregulated, we know that. Remaining hair precursor cells at the base of the shaft will be called as club fiber. Okay. Now, club fiber are only cortical and cuticle cells. They do not have any pigment. The club fiber will gradually migrate upwards with the tricholemmal sac at its base. Now, what is tricholemmal sac? Remember that outer root sheath cell is also known as tricholemma. Okay. So when the hair follicle is moving upwards and pulling the dermal papilla, the outer root sheath cell or the remnants of that shell, outer root sheath cell, will collect at the bottom of the club fiber, and this collection will form a sac-like structure known as tricholemmal sac. So this is the hair follicle, and it is taking the dermal papilla along with it. As it is going up or as it is retracting, as it is retracted, this collection will form of redundant ORS, okay, loose ORS, and this has been termed as tricholemmal sac. Clear? Now, in tricholemmal sac, in the individual cells of outer root sheath, you will just see fibrous tissue in between the cells. So you have cells, and then you will have fibrous tissue in between the cells, and this gives an appearance of brush fiber structure at the level of club fiber in catagen phase. Clear? Let's move forward. Now let's enter the third phase or the telogen phase. The duration of telogen phase is about 3 months for the scalp hair. About 1% of the hair follicles will be shed every day, which is roughly around 50 to 100 per day. Most of the time, we don't even know. Only if we see it lying around on the floor, we'll understand that yes, the follicles are shedding. It begins when the dermal papilla has reached the base of the second hair germ at the level lower isthmus. Remember, in club hair, we ended the Catagen phase when the dermal papilla has reached the level of lower isthmus or the bulge region. From here onwards, we'll begin the Telogen phase. The club hair will lie in the follicular pore before it is shed. When it is shed, telogen phase will be over. This shedding of the hair follicle is known as teloptosis. Okay. Now there can be a lag period. Now remember at the end of catagen phase, all the processes have been stopped. No color, no hair shaft. But still there are some extracellular matrix attachment of the follicle with the surrounding dermal tissue. This attachment will be lost in telogen phase. Okay. Now that period at which in which the loss is happening is variable. So that lag period, that means the end of catagen and the eventual shedding of hair, it is very variable. So this lag period, which is known as between the club hair and hair shedding, is known as the lag period. It's a highly variable, and this lag period is increased in AGA. That means the amount of time the hair spends in telogen phase is increased in androgenetic alopecia. Okay. We also have Kinogen hair. Now, Kinogen hair is telogen hair without club fiber, which has not undergone into anagen phase. Now, remember in hair cycle, you have the anagen phase, you have the catagen phase, you have the telogen phase. Anagen goes to catagen, catagen to telogen, telogen to anagen. This cycle will keep on going. If you have exogen, this is the lag period. But if the hair follicle will undergo again into anagen phase, then this period it is known as kinogen. Okay, so telogen can bifurcate into two parts: exogen if the hair is shed, and kinogen if the hair will not be shed and will enter the anagen phase again. And that is why kinogen is known as true resting phase. Why resting phase? Because the follicle is resting and preparing for the next anagen phase. It is now signaling its stem cells to divide so that you can have a new population of cells which will start the next anagen phase of the cycle. The exogen phase. In the exogen phase, we see an active proteolytic process. That means actively the body is trying to destroy all the connective tissues which was there as a part of older follicle. It ends with the shedding of hair known as teloptosis. Okay. It is a highly controlled timed event in mammals. So in mammals, external phases occur in a timely manner. It can also occur seasonally, and that is true for other animals, not for humans. In fact, in humans, we also have shedding of hair more in certain aspects of seasons. But in animals like mice, you have seasonal loss of hair. Animals like in bacterium camel, which is an example, I think, in Fitz or Rooks, and other animals uh, like you know I'm forgetting the name, llamas and all those animals which Westerners have studied, it's seasonal. There can be an instance which is known as immediate telogen release. For example, if you have taken some medications, or is there any diurnal cycle fluctuation of the body, okay, or if you have gone just from a stressful situation, you can have an immediate telogen release. In this immediate telogen release, the hair will not enter into exogen part of the cycle. Remember, you have telogen, which will bifurcate into two parts: exogen phase, or it will or it will form a competent stage going into anagen, or the true resting phase, also known as kinogen. Okay, but there can be instances when the hair is going into kinogen, some kind of stressful event happens, and because of this stressful event, the hair will immediately gets released out. And this is known as immediate telogen release, which can be easily seen when the body undergoes a stressful scenario. Now there will be a shedding of club hair within weeks of the inciting incidents. Let's move forward. So let's discuss in brief the control of hair cycle. So this is the hair cycle. I've taken this diagram from Bolognia, page 1159. You can go and see the diagram in detail. And a simpler hair cycle is anagen, catagen, telogen, or it can go out by the exogen. So here it is shown as a part of anagen, but actually an exogen is an offshoot of telogen here. From anagen to catagen, FGF5 initiates catagen, TGF-beta, neurotropins, bone morphogenetic protein 25, hairless gene, vitamin D receptors, prolactin, steroids, IL-6, oncostatin. These are all the molecules which will shift the part from anagen to catagen. Inhibitory molecules are like activin, growth-derived nerve factor, insulin-like growth factors. So this will inhibit the transition from anagen to catagen. From catagen to telogen, the, the bone morphogenetic protein 24 signal should decrease. Now remember, from anagen to catagen, BMP24 was increasing. It was stimulating the catagen phase. If you take out that stimulant, if you stop that stimulation, the hair will go into telogen phase. Okay. From telogen phase to anagen phase, that means we have to reactivate hair growth. And what are the genes required for hair growth? WNT, Noggin, Sonic Hedgehog. We have discussed these three. Apart from that, you have hair growth factor, keratinocyte growth factor, fibroblast growth factor 7, insulin-like growth factor 1. So the molecules, for example, insulin-like growth factor, which is initiating telogen to anagen phase, is the same molecule which was inhibiting anagen to catagen phase. That means insulin-like growth factor 1 is one of the different molecules that will increase the duration of anagen cycle, will increase the hair growth. Clear? Steroids will stop or will inhibit telogen to anagen, and we know that glucocorticoids will increase anagen to catagen. So glucocorticoids or steroids are decreasing the phase of anagen. That's why people with prolonged steroid therapy have thinner hair. They will lose their hair easily. Now, protease inhibitors will inhibit from the transmission of anagen to exogen. Remember, exogen phase, even if it goes from anagen or telogen, is an active proteolytic process. That means actively the proteins are dissolving. So all the proteases, all the collagenases are active. If you have protease inhibitor, you will inhibit those proteins and exogen phase will not happen. Clear? So what I'll do is I'll just remove all the markers and I'll wait for 5 seconds so that you can look at this entire control of hair follicle cycle and take a screenshot if you want. Otherwise, you can just go to Bolognia page 1159. Clear? Let's move forward. So after discussing the hair cycle is complete. We'll discuss a bit about hair pigmentation. Okay, how does the color gets transmitted, not transmitted, from the, it has deposited in the growing hair keratinocytes. Okay, let's move forward. Now, we know that the melanocytes are at the upper half of dermal papilla. And the half of the dermal papilla or the bulb cells at the level of the upper half of dermal papilla have hair stem cells. And these hair stem cells will form hair shaft. That means the cells that will eventually form hair shaft, along with the melanocyte stem cells, they are situated at the same level together. And together they are known as hair follicular pigmentary unit. Clear? Here the melanocytes are denser, more melanogenic than the epidermal melanocytes. Melanin transfer is done only to the cortical cells. So the cells that will eventually form the cortex will be the cells which will get color. The medulla, that is why it's not colored. The other hair layers are not colored. It's the cortex which is colored. This process stops in catagen and the telogen phase and it gets reactivated in the anagen phase. Clear? The melanocyte stem cells reside in the bulge region, and these melanocyte stem cells are responsible to replenish hair melanocytes in the anagen phase. So, at the end of catagen, we know that the hair bulge is the only thing left. Anything below the bulge has gone. In the telogen phase, what happens is that melanocyte stem cells will keep on dividing and forming more melanocyte stem cells. So you have melanocyte stem cells, they will keep on dividing and proliferating. They will keep on increasing their number. That is what is happening in Telogen phase. When the follicle will enter anagen phase stage 3, this MSC will become melanocytes of the hair follicle or hair melanocytes. And stage 4 of the anagen phase, they will start to form color or melanin. Clear? So this is stage 3 of anagen and this is stage 4 of anagen. Clear? Let's move forward. Now this is Anagen Hair, and in this picture which I've taken from Rooks page 87.12, you can see that the pigmentation from the follicles, melanocytes, are going to the cortical keratinocytes only. So these are cortical keratinocytes. This will eventually form the hair shaft, and that is why only the hair shaft, the cortex of the hair shaft is the only thing colored. Clear? Let's move forward. No, melanocytes can be seen in epidermis at about 50 days of embryonic gestational age before the follicle has developed, because we know that melanocytes get transferred to the skin first and then the transferred skin will form a placoid which will eventually form hair follicle. We know that these migrate from neural crest to dermis to epidermis. For better understanding, see our video on melanogenesis and embryology of skin. Melanocyte precursors will enter the hair placoid and will divide simultaneously along with the hair fibers. During life, melanocyte stem cells will reside at the hair bulge, populating and dividing their hair keratinocyte during anagen. So melanocyte stem cells, they reside in the bulge region throughout life. And throughout life, they will keep on dividing and forming more copies of themselves. Some of the melanocyte stem cells will get transmitted and form melanoblast, which will eventually form hair melanocytes. And these hair melanocytes are responsible for melanin or color. Clear? While the rest of melanocyte stem cells will keep on dividing so that their population never goes away or never dies out. If the population of hair melanoblast stem cells dies off, the hair will be permanently gray. So you have the progenitor cells, and under the influence of MITF, go and see the video on melanogenesis, MITF and PAX3, the enzymes like dopachrome tautomerase expression starts. And this is the dopachrome tautomerase expression is known as the early melanoblast marker. Clear? So the expression of dopachrome starts, and now we will call the cell as melanoblast. And these melanoblasts will eventually give rise to melanocytes. Clear? Let's move forward. These melanocyte stem cells enter the bulge area during development. After that, they enter a quiescent cycle which is started by TGF-beta. So TGF-beta induces a dormant cycle and the melanocytes will remain dormant there. Notch pathway BCL2, which is an anti-apoptotic protein, these two prevent the apoptosis of melanocyte stem cells. They will prevent the destruction of melanocyte stem cells and they are necessary to sustain the population of melanocyte stem cells. Clear? WNT pathway, again gene required for hair growth, induces MSC to form melanoblast and eventually melanocytes. So WNT is responsible for transmission or transformation of stem cells to melanoblast. Clear? Now melanoblast, they also have different molecules inside them. For example, TRP2, BCL2, which prevents apoptosis, PAX3, which stimulates melanoblast, and melanogenic enzyme, which will eventually form melanin. Low levels of transmission factors like c-KIT, EDNRB, SOX10, LEF, MITF, these are all the agents which are required for progenitor cells. After the progenitor cells has formed melanoblast, there is not much requirement of these molecules, and that is why these molecules are at lower levels as compared to progenitor cells. Clear? c-KIT is required for migration of cells, but now melanoblast is not going to migrate, it's going to stay in the hair, so the levels will be low. Clear? Let's move forward. Now, the melanocyte stem cells are at the follicle bulge. They will give rise to progeny cells. These progeny cells will form new melanocytes in the new hair bulb in anagen phase. Clear? Keratinocytes express FOXN1 and its target FGF2. When keratinocytes have expressed FOXN1 and FGF2, they have labeled themselves that now I am ready to receive the color. And after expression of these molecules, melanin transfer will begin from the melanocytes to the hair keratinocytes. Clear? We know that during melanogenesis, a lot of ROS, reactive oxygen species, develop. If you want to go in detail, see our video on melanogenesis. These ROS levels are highest at the level of precortical zone, distal to the matrix. So just above the matrix, you have the highest level of ROS, and that is known as the ring of fire. So this is just I've mentioned here because I think Fitzpatrick mentions it, so I've mentioned it here. Excessive reactive oxygen damage to melanocytes can lead to hair graying. So if you have a patient of sudden onset, or you could say hair graying, just try and canitis. So just maybe give a trial of antioxidants. And that is why antioxidants work in graying of hair. They reduce the damage by ROS. If the melanocyte stem cells are still present at the hair bulge, the color will come back. But if the melanocyte stem cells have gone, they have depleted, they have finished the population off, the hair will go under permanent graying. Clear? Let's move forward. Mm. The hair cycle dependent changes in hair melanocyte cells. In telogen, the hair melanocytes are in the bulge region of the secondary hair germ, which is the newer hair germ of the growing hair follicle of the next anagen phase, and also in the surrounding connective tissue. But in these melanocytes, there are no melanogenic enzymes. They are dormant. They are not yet activated. They will get activated in stage 3 of anagen phase. In early anagen, hair melanocytes will proliferate and increase in number, and they will migrate in the growing hair follicle. In stage 3 of anagen phase, they will develop TRP1 tyrosinase positive, and when these enzymes have started to develop in the hair melanocytes, it is ready to form melanin. Now, this tyrosinase and all the enzymes will start in stage 3 of anagen, and melanin will start in stage 4 of anagen. Okay. SCF or c-KIT signaling is required for proliferation of HMC and migration to hair bulb. Remember, c-KIT is responsible for migration of cells, so this signaling is required for migration of hair melanocytes to the growing hair bulb. In the Catagen phase, this all activities are abruptly stopped. Let's move forward. So this diagram I've taken from Fitz page 100. This is just to show that in bulge in the telogen hair, only melanocyte stem cells keep on dividing and increasing their number. Remember, in the telogen phase, the hair is preparing for the next anagen phase. So it is signaling the melanocyte stem cells to divide and increase their population. In the early anagen, the melanocytes will go from bulge region to the hair shaft. So now we are at the level of hair shaft, specifically at the outer root sheath level. Till the entire process of anagen phase is over, the melanocytes would have reached the hair bulb, and here the growing is happening. So in the growing hair keratinocytes, the melanocytes will start transferring the melanin, and the growing hair shaft will be colored. Clear? So in the bulge region, you have undifferentiated melanocyte stem cells, while in the bulb region, you have differentiated hair melanocytes. Undifferentiated, differentiated. Clear? Let's move forward. Now, control of hair pigmentation. We know that the process of melanogenesis is same in the hair or skin. There are only slight differences. At the end of melanization or melanogenesis, you have formation of eumelanin, which is brown-black, or pheomelanin, which is yellow. The ratio of these two melanin depends on the activity or the type of melanocortin type 1 receptor that the hair has. Clear? With age, the melanocyte stem cells die off, the hair melanocytes die off, and the level of melanogenic enzymes also decrease with age. That is why you have graying of hair as the person grows. Alpha melanocyte stimulating hormone binds to MC1 receptor, increasing adenyl cyclase, which increases cyclic AMP, and this cyclic AMP increases MITF. MITF induces melanogenic enzymes. Remember, MITF always induces melanogenic enzymes, and after the enzymes have been induced, you have more eumelanin and increased number of melanocytes. So MITF increases cells, increases melanin. Clear? Cyclic AMP increases MITF. Adenylyl cyclase increases cyclic AMP. And binding of alpha MSH to mineralocorticoid 1 receptor increased in a cyclase. So this is the process through which the control of hair pigmentation happens from the higher above centers like hypothalamus pituitary. Clear? Now, alpha MSH and ACTH, these are two different hormones. They interact with MC1 receptor. Along with beta endorphin, this is another hormone which acts via mu opioid receptor on the hair melanocytes. These two pathways are known as pro-opiomelanocortin pathway. They promote melanocyte proliferation, dendricity, and pigment production and transfer. Mu opioid receptor also regulates hair pigmentation via protein kinase C beta. Negative signals, that means signals that will stop color formation, include brain and muscle ARNT like 1, cortisol, and PER1. So I'm just mentioning it here for academic interest. It has been mentioned in Fitzpatrick, so I thought I'll mention it here. Okay, let's move forward. So we have the proteins that will increase the hair color, and we have the proteins which will decrease the hair color. Clear? Let's move forward. So with that, we have finished the phases of hair cycle, the control of hair cycle, hair pigmentation, and the control of hair pigmentation. I hope I have made the hair cycle simpler, at the same time being comprehensive to understand. These are the reading recommendations. Otherwise, this video should be just enough. Again, shameless plug. Do consider channel membership. Only 10 day, roughly 8 days are left in this year, and it is just available at 59 a month. We have newer videos. The videos for members are longer as compared to the other videos which we give it for public, and those videos are more extensive, more detailed, and will help you in developing an understanding of the processes. So if you have gone through this video, you'll understand what I'm talking about. So with that, I'll finish the video on hair cycle and hair pigmentation. And I hope that after this extensive video lecture, you're able to have a certain understanding of the process of hair cycle. How does the hair goes from anagen? What all happens in anagen phase, then in catagen phase, and in telogen phase? What initiates the shift? When do we say anagen? When do we say catagen? When do we say telogen? And how is the color of hair shaft is controlled, how the color actually eventually develops and transmitted to the growing hair shaft. All of these things have been covered in this video lecture. I will not take much time. Mention any comments and suggestions in the comment section, or you can email me directly. Till then, adios and bye bye.